Assalamu alaikum wa rahmatullahi wa barakatuh, everyone. I hope you're all doing well. So, I'll do another round of attendance afterwards, but in the meantime, get yourself ready. Make sure to wash your hands. Place your belongings at either a coat rack or in your cabinet. Make sure that there's nothing obstructing the bottom of the restaurant. So before we begin the lab quiz, the pre-lab, I'm just going to go with another round of intros for attendance. So just to bear this in mind, when I call your name, just say here or just raise your hand. I'm just going to see numbers. Okay. So there may be people outside. I'll just check and then we don't have to begin the lab quiz doesn't check, but if ever you get, then wait for the time for the So because it's time reminder, you don't have to begin the pre-lab quiz yet. But if you have left your backpack or any belongings underneath the coat rack, this is your time to place them inside the cabinet. Very important. You don't have it, and it shouldn't be that there's some gases in the belongings here. So I would highly urge you to place them in the cabinets. There should be enough space. So I'm going to go with attendance once again. And once again, you don't have to do the two lab, but you will be, and I actually want to make sure everyone left with the bottom, at the bottom of the floor, time to take. It is 2 to 3 times, and both on the floor. Please don't leave it there. You don't want bacteria really on your belongings, but also there should be enough space for. Anyone else left their belongings on the floor? I see a checkered red and white platform. And I see please make sure whenever you come into the lab that you place your belongings in the most appropriate location. And hopefully you've all seen my Quirkus announcement yesterday, but this is something I really want to reiterate for everyone's success in the lab. And also on that note, I know a lot of us may have bulky bags, but if you're able to either get a locker or condense it when you come in, that would be easier for you to be able to place it back inside the cabinets. Otherwise, technically speaking, you're not actually allowed to place anything above the cabinets for safety protocol, for safety reasons. And if that does occur for the upcoming weeks, then that's where things may be removed from either your backpack, or you may have to go through additional protocols. So just bear in mind, everyone, don't forget, I'm going to turn on the mic, but don't forget, everyone, the importance of making sure that you have only what you need for the lab. Hopefully that makes sense. Any questions on that note? No questions? Okay, so just remember once again, next 2 weeks from now, 5 weeks, just make sure that you have your belongings. So now, with this in mind, let's move straight to attendance. And for those that don't know, my name is Muhammad Saeed, and very grateful to be your TA for BIOA02. I'm also TAing 39, so the Wednesday of last week was my first practical for AO2, but nonetheless, we're going to have amazing fun with each other, inshallah. So moving forward, I'm going to just call people by names, and if you find your name absent, just let me know. I'm going alphabetical by last name. Starting off with Bill Abad. Next is Andrea Ahumu. Can you let me know how you pronounce your last name? Ahmada, okay. Thank you. Aaron Kramer. Okay. Okay. Jai Sheng Fu. Jai Sheng Fu? Or Fu? No? Okay. Jizian Gao. Yes? And did I say your name correctly? Did I say your name correctly? Yeah, okay. Feel free to correct me. Salma Garcia Rodan. Garcia. No Garcia? Okay. Eva Jill. Okay. And okay. Nicole Joseph. Okay. Nicole Joseph. Karis John, or John Karis. No. Okay. I have an updated list, so I'm just going to go by it then. Mahir Hantush. Here? Okay. Mahir? Mahir. Okay. Giselle Haye. Giselle Haye. No, okay. And then John Karas. Did I say John Karas before? No. Okay. Faizan Chef. Okay. Uh, so I think the attendance was modified. Okay. So if I missed your name, just let me know. John Karas. Did anyone go for John Karas? Anyone here? John Karas? No? No? Okay. Tanika Manuharan. Yes? Okay. Okay. Charlotte Mu. Zala Nasari. Or Nasiri. Okay. Bishop, I think I saw this person. Yeah, okay. Noseworthy Ralph? Yep, okay. Ashwin Nagopal. Forgive me. Joshua Shank. Okay. Miriam Simaga. Okay. Selena Singh. Ranran Sun. Okay. Andrian Tang. Okay. Okay. Nafisa Tasneem. Okay. And Vishnu Mohan. Yep. Okay. So I have about 4 more people whose names weren't included in the list. Can those people please raise their hands? Whoever I never mentioned in the attendance. The reason being is because we have 2 empty seats here. Who did I not call? Did I not call your name? What's your name? Can you spell out your last name for me? Zhou. Can I see your increment? And anyone else, because there are 2 that actually should be 1 more person who I didn't call. I don't want to be doing anymore. Believe my call. Did I call you? Anyone else? So just bear this in mind. I think because it is the first week, some things may need to be adjusted, but hopefully if there's any concern, let me know. I'm just gonna also adjust the audio. Oh, okay. So hopefully now, yeah. Okay. So once again, my name is Mohammed Saeed, and I'm very grateful to be able to be here with you. I have some experience in research. I graduated here in Cell and Systems Biology, August 2024. So I've been like in the works of at UTSC for some time. And very, very excited to teach this course since the time I took it maybe 7 years ago. So there's things that have changed, of course, but hopefully throughout the duration of today's practical, you'll be able to see how you can connect the dots with different concepts, lecture material, and integrating it into your own life, hopefully. So moving on, and I mentioned a few lab safety rules. Also, how many of you have seen my inbox or Quirkus announcement? Please raise your hands. Okay. So about half of you, maybe 54% of you, I would recommend having a look, see that in case your notifications are off on Quirkus, to make sure that it's up to date. So that way, if I send announcements early on, you'll be kept in the loop for any post-lab and pre-lab announcements. Now, in terms of safety material, I'm just going to go briefly to this. And pretty much We have a lot of equipment, as you see it on your benches. Sorry. And pretty much a lot to take into account beyond and within the lab manual. And so as we go through presentations, you hopefully will be able to understand what you need to do throughout the duration of your lab. We have a lot of apparatus for the exercise 4 shown here, exercise 2 and 3 there, and hopefully the materials that you need will be able to find. You also need to microscope size proposal on your benches, and I'll be separating with one. Just gonna check if things are working. So, and as you come in, wash your hands, right? Wash them with soap. And for that, we do something in your eyes where the eye wash station is. I don't really want to turn it on in this water, but the idea is that you put it this apparatus, here, then automatic will fly. If for whatever reason you have flammable material on and actually catch it on fire, hopefully that's not the case. You spin it around, water goes down, shower as well. Which is why I just want in general us to be very mindful of space. Not to please leave anything like coats or any packaging items on the ground, but rather into the appropriate spaces. Here for your coats and there for your bags. Ideally, we should place, and there should be enough space for bags, even if they're big, because these are quite deep. So you should be able to place your belongings inside. And if needed, you can always take out a few items so that way they can all fit together within a single location. But just bear that in mind. For upcoming weeks, we can actually do this. And personally, I don't want anyone getting in trouble. So just bear that in mind. In terms of, and if there's any questions, for spills, actually clean up and will be, if for that reason, I'm sure you have to clean the room here. So that very edge right where the coats are. And if there's any need for cleaning off glass, or if there's glass that are used, then we'll replace the generally speaking, you don't have the necessary items for you to be able to know from your manual what to use and what to clean up quickly. So just bear in mind any case reminders or pieces of advice that you give over the box. That way you're able to see throughout the duration of the course. So that you're getting to that. Any questions? Generally speaking, oh yeah, material. There's the top right there. There's 2 computers if ever needed. And of course, my food or drink should be clear or at least being used. The lab cell phones as well, so that you don't need them. Sharp objects, be mindful that there are objects that are here, for example, that like scissors are to be returned back to their appropriate location. So just bear that in mind. Many of the instructions are already here for the lab. You just have to be cognizant of what's needed and how you can be most proficient and most efficient in the duration of the practical. Any questions? Okay, so let's go straight away to lab 1. In terms of the duration, rather than having the pre-lab quiz earlier, we can actually begin it if you haven't done so already. So what you could do is begin the pre-lab, but bear in mind a few important points. It's going to skip. Okay. To here. Just to reiterate this for everyone, and hopefully you're able to see this, the quiz, as you're aware, should be done individually. But if you need any items like your lab notebook or your lab manual, a calculator, any materials that are needed, you can make use of them. Bear in mind that we start in the beginning of the first 10 minutes of the lab. So punctuality, as I mentioned, is very, very important. Bear that in mind, everyone. And yes, actually, in the quizzes module, of AO2, you'll be able to find the pre-lab quiz. So begin it, and hopefully you'll be able to excel. Any questions? If not, this is your time to continue with it. And I think, yeah, you can begin. Once again, you will not be allowed to interact with other members, any bench members here. But just make sure that you complete your lab quiz. If you need a computer, there is computers on either side. So if you need it, it's okay. It's 100% acceptable. Just bear in mind though that you need to complete the quiz at the first 10 minutes of the practical, excluding for today, but in general, 10 minutes onwards, you have to be able to complete the quiz, and it is timed. So bear that in mind, everyone. Any questions from anyone? Oh, yes. Yes. Computers are open. So just a reminder in terms of a few action items that I've said and recommended for all of you. And I want to see you all succeed in your journeys, be it in research, academia, medicine, and any professional or any career option or journey that's best for you. But just bear in mind a few important points that I try to recommend the following to students. Amongst them is discipline. Like trying to, really focus on, and with punctuality, try to really focus on understanding the concepts with integrity, not being too lazy or flipping through your phones, because I can keep an eye and keep track of this, of course. But in terms of integrity and authenticity, those are really important points that I hold dear. And so I hope everyone will feel comfortable, especially throughout the duration of our practicals together, to make sure that if they feel that they aren't able to balance, let's say, those traits that I've mentioned, punctuality / discipline, or integrity / authenticity, then just let me know. And one other important point that I do suggest for everyone is critical thinking, right? Because you're in a lab and there's a lot of things that you could connect the dots with. In fact, what I'm going to try to do is in exercise 4, touch upon a technique that's used for protein expression. So hopefully you'll see and hopefully connect the dots with different upper year concepts that you'll learn down the road. Any questions? How many of you are complete, or actually, how many of you are in the midst of completing your lab quiz? How many of you are doing it now? 1, 2, 3, 3. Okay, so maybe about, I'm guessing maybe in 5 minutes we'll be ready to continue. But let's see. In the meantime, there are things that you could think about. Look at the law manual. Refresh yourselves on what's to be done. To make things easier, I'll wait until 33, and then hopefully you'll be able to work as groups. Oh yes, sorry. So maybe just in the meantime, separating the blocks before, 3 to 4. The idea is that, and I know this is only of 2, but maybe I should move 1% accordingly. For now, and then you'll be 1 block instead of 4, and then very much everyone that in this lab, I want to make sure you all excel and do your best. So just bear in mind that you're paying attention. One of the other tips I gave for the practical. So bear that in mind. In about 2 minutes, just to be mindful of our students, right? For those that weren't able to access the quiz, that will start in about 2 minutes time. But think about in terms of the order of today, things are different from the lab manual from sections 1 to 4. Instead, we'll be going from exercise 4, which is what's going to prepare us for the formal lab report, to then exercise 2, which is going to be looking at those flowers right there, and then cutting them into halves to take images or to draw in our lab manual. And then exercise 3, we're doing more statistical analysis with the pollen grain diameters found from the slides here, or even from the pollen of your group, which we'll discuss briefly. And then we have cleanup as well. And that should conclude the lab. Yeah. Instead of placing, and once you guys are done, we have different items. I would just say it's very important to also keep in mind organization, because you're going as groups, but there can only be really 2 people per group that come in to that given location in the middle bench. And so during that time, try to segregate as groups as to what should I do and what should my peer do. So that way things can be more efficient, and hopefully you'll be able to call it a day in the 2 hours. So that's the plan. Now, any questions from anyone? No? So I'll go forward with the practical for now. And then if anyone is still doing the quiz, just don't worry, you can continue with paying attention and learning from your group members shortly. So let's start off. And if there's any questions, let me know. So exercise 4. Let's get ready. Okay. So let's have a look at your lab manual as well for reference. If you go to exercise 4, you'll be able to see that the following, right? Your mission, essentially, your first experiment of AO2. And that's where essentially you're going to be preparing a container, and I'll show you those containers shortly. But essentially as groups, as the groups I mentioned earlier, you'll be separated to take on one of the following treatments, and each of them will be replicated, so you don't have to worry about doing all of them, but you only just do one category. So bear that in mind, and then essentially the goal is to test the effect of drought, because we're doing different treatments. Does anyone know what a control treatment is? Yes. Yes.
What you're for assessment everything for perfect circumstances, and that you're crucial.
Generally, yes, yes. And so actually, great answer. Can you remind me your name? Josh, last name? Shan. So like it's very important that that when we're doing experiments, we also have controls, because if you ever make a mistake, for example, you'll be able to cross-check with your control to see, oh, maybe my control isn't working out as much as I expected. For example, our control here is no drought. So if for whatever reason you saw that there was dwindling of your plants over time, then that may make you suspect that there could be something wrong with the experiment, right? Because you've given them enough water and you've watered them, as you can see, every day. Whereas the other experiments where we're doing or testing or stimulating for drought, there's the case of treatment 1 where you're watering every other day, right? And so there is a day in which there is drought or at least limited water provided or supplied. And then drought treatment 2 is where water is only provided every 3 days or every 3rd day. So there's essentially that's something that you want to keep in mind. In terms of setting up the experiment, actually, before I move further, I'm going to separate you now into groups. So, and just bear in mind your name for the group, so that way you'll be able to label it for the tape and your container. So you'll be control treatment A, you'll be control treatment B, you'll be treatment 1A, treatment 1B, and then treatment 2A, and then treatment 2B. So just bear that in mind. I can go just to show you once again. Your control are treatment 3B, treatment 4B, then control treatment 1B, and then treatment 2B. Now, I'm just going to go back there. Control treatment A, treatment 1A, and then treatment 2A. So bear that in mind, because essentially we have different, and I'm just going to skip slide. If there's any questions, also let me know. So setting up your experiment is where you start using advanced materials. So for example, you have containers for each group, 7 groups. So bear in mind that we'll see these large long balls essentially of treatment each water. So you have treatment 1, your will go actually into the sun. I think it's two. So the two edges will come to that side. And then control treatment, stage one, again, we just put this first below. So the idea is, and you see the different papers shown here, and like the one I have on the slide, which I'll keep there for now. But the idea is that it's very important that you follow the instructions. Yeah, of course. So the idea is that you pay attention to the instructions. I'll show it as you can see here. The idea is that you have a container, like the one I showed here. And when you're starting off is with taking half a piece of paper towel, pretty much just rip it, you place it inside on one end, so that will be remaining as even and as thin as possible. You don't have to squeeze it per se, just make sure that it's particularly even. And then, so once that occurs, you just place half a piece of paper towel. And this is a good simulation for those that want to know, with the label as well, for cake. And then you have cotton. Cotton is here, and you'll also see the amount of cotton that you need to add. You add cotton, just like cut a piece or cut a layer of cotton, and then you just add that on top, and then add another layer of paper towel. Bear in mind that you're adding in water, I believe afterwards. And then so just bear in mind where it's located in the lab manual, but think of it like an ice cream sandwich where you have paper towel, cotton, and paper towel, and then you've added water, I believe it's 30 mils. 30. And the instructions are also here, which is nice. And so 30 mils of water, and how you're doing that is using the larger graduated cylinder. So you add the water from here, just make sure you don't spill, add in 30 mils, move slowly, take your time, but don't also delay. And then after you've done that, place it on, try to make it evenly distributed, and then you continue on with adding your seeds. The seeds are going to be with the small graduated cylinder. It's going to be 10 mils. 10 mils of the seeds. So I know these are solid, right? But you're going to be adding them up to 10 mils. So bear that in mind. Not 10 seeds, but 10 mils. So you're going to add those seeds. You're going to layer them up. So think of it like a cake, right? You're going to layer it up such that it's going to be evenly distributed with seeds. Don't have them centered just in the middle, but have them distributed across that layer. And then afterwards, you're going to add in one other half layer. It's going to be a second sheet of paper, but another half layer of paper towel at the very top. Any questions? Once you're done, and bear in mind, you can even begin labeling the seed. If you find that another group is already busy taking care of the treatment for their rapid, you're going to be actually placing them not there, but down there. And I'll show you where exactly, but the point is you're going to be on the left-hand side, in here, and placing it for week 2, Wednesday, and essentially the timing and week that we are all here today. So just bear that in mind. That's exercise 4. We can be done this, I would hope, by 10:50, and that shouldn't take too much time. Any questions? Okay, so you're on your way. You can get ready to go. You can go and just remember your name, your group, and you can move forward now. What's your name? Yeah. I have the. If you have any questions, let me know. If you also found that I didn't mention your name for attendance, also let me know. Thank you. Any questions? Oh. I think just as long as you make like a certain. Yeah. So. Also, once you add your seeds, afterwards there is another layer of water that's being added. Just bear in mind the location. When you're done, adding in your final layer of paper towel. If you have any questions or having any trouble, just let me know. Okay, water, 30 mils. Yeah, they have anyone. No, we need the big one. Oh, that is, wait, is it? Oh, yeah, that works. No, never mind, it's 25.
30 milliliters.
No, I'm just sort of. Trying to make sure I understand all the steps and everything.
Okay. Do we have it?
How do I do this smoothly?
It doesn't have to be like that. This was 30-ish. Perfect. Just pour that in. But yeah, like on the sides, yeah. Seeds, 10 milliliters of seeds, I think. Yeah. You saw that small thing? Oh. Yeah. Any questions? Any group having any problems? Let me know at any time. When you're pouring in the water or the seeds, try to have someone hold the base so in case it doesn't fall or spill. You can, what you need to do just before you're done, and just bear in mind that you've added in the last layer of water. Once you've done that, you can label it with your group. So your practical or practical 40, your control or treatment group, and the replicate letter, and then the dates as well. I think another thing to tell, right? Yeah. And I think there's 20 more milliliters of water.
Exercise 2. Is your name?
Just this is a week 2.
Yeah. Okay, so I should probably all those pages.
I know. Yeah, but that's good. Great question.
Just making sure that up to date. So
That's great. Yes, so for those that aren't aware, we'll go back to exercise 2 shortly. We just want you to be able to complete this first.
Yes, just by that project of 42A.
And then also when you're done, label it with the date as well. So it helps coordinate timing for you. The date is, I believe it's 15.
You want with the date?
So what? Yeah, 2025. Any questions? Anyone having any problems? Do you think we could be done in the next 2 minutes? All right, is your group done?
Yeah. Okay.
Okay.
Yeah.
And then, for example, if you're treatment 2B, then you could say T2B as a nice acronym.
Okay.
1/15/25, which is Jan 15. Once you're done, you can place it back in the purple box. I would recommend that they're dry, so just maybe just air them out. If you could just also I'll show you. When your group is done, make sure that you place back the cylinders and any other items used back into the purple box. So that way it cleans up and keeps everything organized. So how I'm expecting is that there should be 5. Then you can great job, you guys. Great job. So we'll wait for about 1 to 2 more minutes, and then we'll move forward. But what I wanted to say about this and how it connects, like integration, it's a common theme that you want to be able to connect the dots with different fields, different topics, and ideas. Here, when it comes to like setting up this container, it's similar to an analogous with Western blotting, which is a concept when people try to, like biologists, explore protein expression. So what you're doing is like similar to what's in a gel electrophoresis array, is essentially aligning up different layers of, like, it could be film, essentially styrofoam, and other things to be able to essentially coordinate an efficient growth or an efficient transfer and essentially delivery and signal of your proteins. And in a similar way, I mean, what we're doing here is building up different layers of, paper towels, cotton, etc., to be able to eventually grow out these seeds, right? So if you actually look at the left-hand side, you'll notice those are actual plants, some of which actually include my practical. So, but nonetheless, the idea is that your plants, the ones that you just built right now or have sown, the seeds that you sow right now are the same seeds that you're eventually going to see grow up down that side. So, like, I mean, they grow at different levels, right? You can see how perhaps like you come on here later on, you notice how different growth patterns exist with the different effects of drought, right? So keep that in mind. I mean, this is something that you're going to see in 2 weeks' time for your formal lab report. So just think about this, think about it a bit, and see how hopefully that should be beneficial for you all. Exercise 2. Any questions before I continue, by the way? No? Okay. So exercise 2, you could still stay in those groups that we've had from before, but now what you're doing is actually taking these petals or these flowers, the lilies and tulips over here. So you'll have 6, yeah. So if you want to take either the purple or pink flower, and essentially you're just going to take it out. You're going to be trying to essentially on the first figure of your lab manual, draw out how your flower looks. Right? So there's a difference of theory as you see shown there for the apple flower versus reality and actual practicality. So these are the tulips, for example, but that's an apple flower. How do they resolve different, features of the flower? Is something that you should consider because that's what you're going to be drawing out. And hopefully when you're drawing them out from the external view or essentially what it looks from the outside and even internally, for your first figure, you'll be able to draw out these different components. The sepals, which are underneath, as a consider them like petals, but they're underneath. They're the outermost whorl, essentially, or feature of these flowers. Leaves of these flowers. Technically, they're not leaves, but the point is that these are the first whorl is at the outer edge, and that's what sepals are. Afterwards, just a bit more up, you have the petals, and then after that, you have the stamen. That's a male reproductive system, and then the pistil, which is the female reproductive system. Bear in mind that there are different numbers, right, for the male versus female organs, right, and they have different compositions, right. And so the idea is that for the case of like the stamen for males, you have the anther, which is at the top, which is where the pollen comes, and then the filament is where there's that exchange of nutrients in between the anther and the flower itself. And then within this angiosperm, right, these are essentially plants with flowering plants, essentially. These types of plants have as well their pistil, which is essentially composed of the style at the top and the stamina at, I believe, stigma. Yeah, stigma at the top, and then the style at the very length, essentially at the very edge, at the height. The height at the very top is the stigma, and then along with the pistil is the style. So just bear that in mind. You'll notice a pattern as well. If you go from top to bottom, it's SP. So that hopefully helps you understand the different concepts. I'm just going to go briefly to let you know that, and the reason I was saying about these flowers, like they make up different abundances in the population for their own distributions. So monocots, which are sets of 3 petals / sepals, versus eudicots, which are more abundant, but they make up sets of 4 to 5 petals / sepals. And that's pretty much what I've just said right now. And that includes actually pretty much the organs that I've said. In terms of their morphology, which is something you want to bear in mind, right, when you're writing your figure caption, just like the monocots versus eudicots, that you're able to understand their symmetry. So, for example, what you see here is a eudicot, 5 petals, for example, and you'll notice that they have a radial symmetry, wherein if you were to cut at any location, think of it like a pie or a pizza, then you'll be able to see that there's general symmetry across all sides. Bilateral or zygomorphic symmetry, and that's The radial is acidomorphic or actinomorphic symmetry, but the bilateral one is what you'll think about as like a mirror image. Like think of it as your two hands, and that's pretty much what you have of some plants as well. So what you're going to do really for today is make use of your microscope, right? If you, as you essentially look at your flower, you can look at it under the microscope, the dissecting one that you have in front of you, and you'll be able to outline the different organ features of the flower that you have, as well as the properties I mentioned earlier, their symmetry and their trait as either eudicote or monocot. When you do this, make sure that you include in your figure caption the details of the flower, including the symmetry and property of eudicote versus monocot, but also bear in mind the annotation or labeling of the six or eight features that are shown here, from sepals to style. So let's spend about, I'm going to just check the time frame. I would say this should be done because this is half of it by 11:20 or even 11:15. So try to be done that component that I just mentioned by 11:15. And then we'll go to the second page or the following slide, which will take a bit more time. Any questions before I move forward? And before you begin with taking annotations or images of your flowers? No questions? Okay, so let's go straight ahead. And then after you're done or around 11:15, I'll show you what's next for your second figure. Thank you, everyone. So just come by with your groups. One person just has to grab a flower, and then eventually they could just open it up, and all of you draw out for yourself individually the flower in your figure. Remember the different traits that you see here: the sepals, petals, stamen, anther, filament, pistil. And I'll show you the previous slides as well, so that way you'll be able to see it. You don't have to open it too much because you're going to be dissecting it afterwards, but the point is try to open maybe one or two leaves at a max, or one or two sepals / petals, and then hopefully you'll be able to draw it out.
Yeah.
Any questions?
I was just thinking about how. Yeah.
So what if like,
It's actually kind of a good thing.
Any questions, anyone? Do we know what we're doing? Yeah? You can for now, if you're able to.
Right. Okay, yeah.
If you want to use the dissecting microscope, you can, but I would recommend at least trying to use it definitely for the second component. Anyone having any questions?
That's the.
If you wish, if it's difficult, there's two options. You could take it as like an eagle's eye view, like the top view, and draw it accordingly. Or you could take it from the angle as you see it, and then draw in the internal parts as if you're seeing through like an X-ray. So you have both options. It doesn't have to be too difficult, but definitely try to incorporate all of the components or the 8 components I mentioned earlier. That includes the sepals, petals, stigma, the pistil, and the stamen and pistil, and then the different the 2 inner components of each reproductive organ. Hopefully about 5-ish minutes should be okay, because the idea is that you want to be able to at least draw each person for about a minute. Each person should at least hold it for about a minute, and then the organs themselves can be available thereafter.
This started with so
Yeah.
I think that's fair. Yeah.
just because we said it was 5, I'm saying. Yeah.
Just another question or thought for you all. You maybe saw from your manual the word tepal. What's a tepal? Does anyone know what a tepal is? The tepal is separate from the sepals or petals. In fact, they're almost indistinguishable. But just bear in mind that you're not looking at tepals as a word or as a term in your figure, but you're using petals or sepals. Sepals are underneath once again, and you could compare that with the apple flower, right? Here's the example right here. Whereas the petals are on top. So think about if you're able to distinguish the top versus bottom leaves. If you can't, then that could indicate, or I would recommend actually finding out from your flowers which components are above the other. When you're ready, when your group has done taking the image, so just bear in mind, everyone, each lab manual needs to have both figures. The first one that we're doing right now, and then the second one is when we're going to do the following. So go back to the following slide. So just in the next few minutes, try to be able to complete the first figure, and you can also, if you're done this, make the caption with what I've mentioned thus far: the monoclot versus uniclot, differentiation or categorization, characterization, and then the symmetry as well. So think about it. If it's a syntomorphic, it's radial. If it's bilateral or zygomorphic, then you'll be able to distinguish the two. The idea of it being eudicote or monoclot is influenced by the number of sepals / petals of your flower. So just think about those properties. If you need help of going back, or you can look in your manual, but if you need me to go back to any previous slides, just let me know.
Wait, where did it start? Oh. Yeah.
So I would recommend not taking out the leaf.
We did, it's like bad on us. Yeah, but yeah.
There's might be also really proud Still have water.
We did it pretty good. Everyone else felt
Yes.
the next one. Looks like they don't really- I think like the petals are much thicker and wider, whereas the sepals are more thinner or more curved and smaller. So if technically it's only the sepals, but if you were only to look at their essentially position at the very like scale range, but just if you're focusing on the position, and you would be able to at least look the ones that are above or petals. Otherwise, you can refer to them as. Any other questions? Does that help or? Anyone? Yes. What's your? Okay. We get it. Still have both properties labeled, still have petals and sepals labeled. If your flower is being split and is hard to distinguish, but at least mention them both, and if you wish, you can include petals as well as like a hybrid set of leaves or pieces of your flower. How are we doing for moving forward? Can we move to the next step?
Are we ready to move to the next figure? Yes or no? You guys look like you're ready? Yes? Are you guys ready? 2 more minutes, okay? 2 more minutes for this side as well. Do you think you'll be ready in about 2 minutes? Okay. And human 2A, are you guys done? Yeah. So remember everyone, I'm gonna have to go quickly to the second figure shortly. If you've drawn your figure of your flower, please make sure everyone, I hope you're paying attention. So please bear in mind for your second, for your first figure, like your second, you're gonna need to be as descriptive as possible. You need to refer to not just generally the labels of the structures that you have, but also in terms of what the flower is. Remember the symmetry that I mentioned, and also if it's udicot / monocot. So bear that in mind. The labels are already mentioned in your diagram, but just make a reference to the fact that what type of species you're investigating. Is it a lily? Is it a tulip? And you'll know the genus hopefully. In the genus for the tulips is Tulipa, and the lily for the lilies, the genus is Lilium. So you can write that down for your figure caption. Tulip is Tulipa, and lilies are Lilium. So just make reference to that for your figure caption because it needs to be as descriptive as possible. Always, whenever you're in this course and any upcoming courses, generally speaking, your figure caption needs to be as detailed as possible, such that you'll be able to understand what your figure is illustrating overall and comprehensively. So please bear that in mind. In about a minute, or actually in less than a minute, we'll move to the second slide. Any final questions on this figure? No? So I'll move on to the second component of today's exercise 2, and that is essentially now cutting your flower into half. So the idea is that there should be a razor blade in each component of your groups essentially. The razor, so you have a petri dish which you have to cut. Actually, sorry, you help visualize your flower under the light microscope, the dissecting microscope, but you have a razor blade here to be able to cut and tweezers to anchor your flower. Please bear in mind, everyone, that when you're using these, especially the razor blade, try to keep it at the widest end of your flower so you don't run the risk of cutting yourself at a very thin level. Bear in mind that these are obviously blades, right? So you have to be careful, right, not to cut yourself. And then you're using both the tweezers to anchor on your plant or your flower on the widest edge, so you have more room to cut. And you're using the razor blade to finely cut through the not through, but just the styrofoam as your anchor. So just bear that in mind. Once you've cut it, you have 2 things that you can do. You can place it under the petri dish, which opens the dish that you see here, the glass one. You open it up, place your flower on it, and then you view it from your dissecting microscope. And then every member of your group can look at it and take turns accordingly. And then when you've done that, you have the option, and there should be slides. Miles, do you think they have slides here? Slides for the on paper. Perfect. Yeah. So slides. Now, when you're done with visualizing your second figure, or visualizing the internal pores for your ovules and the ovary of the flower, right? Because you're dissecting, that's how you find fruit set features of your angiosperm. Once you've done that, you'll be able to take the pollen and move them onto the slides. Further analysis for exercise 3. That may have sounded like a lot, but a good question.
Any questions? This one? You could definitely use it over your hand. I just wanted to make sure that.
Like, yeah.
So you could cut it, you don't have to cut all the way past the stem. What you really need to do is make sure that you're able to cut all the way to, I would imagine, up to here. So you're a bit past the ovary, but that way once you open it up, you'll be able to see the ovules as well. So just bear that in mind, everyone. Any questions? Yeah.
Might as well. Yeah.
A bit hard to see the ovules themselves, but you'll notice a different tone of color. That actually helps us indicate that these are the ovules. So just bear that in mind.
Okay, yeah.
Once you cut it, you can let me come by. I can have a look at them. I just want you to make sure that you cut neatly and nicely and finely, such that you're able to cut through without shearing off your petal or your stem. Bear that in mind. It's possible. Any questions? So once you're done this step, you're going to be doing your second figure, or once you've cut the angiosperm and flower in half, you'll be able to then do once, and you can actually, I don't want to say this, but it's just more like a surgery operation, so you hopefully be able to understand it down the road. So you can use these needles to poke through the leaves to help anchor on your flower. If you don't have any more petals or sections of your flower, you can also poke through the stem. That way you're able to anchor it well, and then you'll be able to cut the main.
No, it's the experience. Like, there's no. Like, when I was looking at it. Like you can see it better by like naked eye. That might be too much to that. But that's my job. Yeah.
I cut too much to one side and I like.
Yeah.
You can go, but yes, just a bit above to compare the state of find like small ovules. You'll see it between your Amen.
Try it. But you don't need anything else.
Yeah.
Yeah. Yeah.
It's a bit hard to speak. They are different. Yeah, but it's, and that's interesting as a whole, like, you can see. I think we could go by, right? No, it's not to look at the technique or like, look at the values, right? Or actually look at the place where it's down there. That's really so pretty much around there. It's harder to see, so obviously it's hard to see through, right? You're dealing with a flower, this isn't like a simulation. So just bear in mind that's part of the protective layer and the design of a flower, that it has the ovules much more secluded within the ovary. And similar, like to think about your, like reproductive organs, like other, species, and just think about how applicable this is, right? It's like if someone were to, try to do other mutative experiments, like how difficult would it be to actually reach the ovules with something that you could consider, right? So think about these types of things, interpretations, implications of your findings, because it helps you gain greater knowledge and insight, not just into biology, but into reality. So hopefully that helps. In the next few minutes, we're actually going to have to move to I think the second last experiment, or the last experiment for today. So just bear in mind that we're able to finish drawing out, once again, the 6 or 8 components. I know it's a lot, but it's honestly, if you're able to draw it out, or at least write out the components in the layers, it shouldn't be too difficult. It's pretty much the 8 that I mentioned before, plus the 2 that you found internally. The the ovary itself, right? And then the ovules, the ovule with the eggs inside. So the ovule is the one that I mentioned earlier. Sorry, that's a thin white line. And then within it is where your eggs are. So just bear that in mind when you're drawing it out. It's pretty much you have the ovary itself, then a thin layer of your ovule, which then includes your eggs, the eggs for the plant. Yes.
I don't think that's the light.
So the ovule is structured. So the outermost is your ovary, inside is your ovule, and then within your ovule is the egg. We just are asked to say ovule plus eggs, and it's hard to just create them together or separately. But the point is that you have the ovary that you have to include, and then your ovule, or ovule plus egg inside.
So,
Yeah. So we pretty much what we see.
yeah.
So that's going to be all year. Outside that, city, and then we did all you.
Okay. Yeah. Okay.
Okay.
It makes it look. Yeah, great.
So, just I want to recap with everyone. Let's see timing. So actually for timing, it may be tough for me to go through this once more, but everyone, I want to make sure you're paying attention. I mentioned this earlier, but I'm just going to say it one last time. Make sure when you're doing this annotation that you're having a descriptive figure caption, that you've labeled the ovary and ovule on top of the other features I mentioned earlier. The ovary, once again, is like this structure right here, but the ovules are internal. Sorry, the ovule is an internal structure housing the eggs. So just think of it like that, right? You have like the bricks outside of a house, the inner foundation, or think of like the pillars, for example, and then within it is your appliances. So the idea is that you're having almost three different layers, internal structures that could be from external to internal structures that could also be included into this figure caption, into your figure. Any last questions before I move further? No? Okay, so we're going to move to the next step actually. And this is where we're going to lead go to exercise 3. So is everyone on page? Like, is everyone aware of where we are right now? Like, can everyone go to the lab manual and see exercise 3? Okay, so now just so everyone's on the same page, you have pollen right at your disposal right now, but the idea is the following. I referred to the statement earlier, right? The male reproductive organ, and so within it you have that pollen that's being produced, and you have it undergo two different stages of cell division. First off is meiosis, right? So you have the sporangium that has multiple haploid spores. What's haploid for those that don't know? So haploid is referring to the number of gene copies that the spore has, meaning in this case it's N, so it has just one gene copy from the plant itself or within the stamen. And then you have mitosis that occurs following spore formation to eventually lead to the development of the gametophyte, right? Hopefully you've learned this by now in your lecture with Ivana Stelik. But if not, the idea is that the gametophyte is going to develop, right, through mitosis or following mitosis. And the idea is that it's eventually going to mature. It's still as at one end, right? Because the idea is that or at least presently for the time being, the number of gene copies of the pollen is going to be 1 as is. But following, fertilization, where the pollen reaches the egg, that's where eventually you're going to have fertilization, and then it'll lead to the completion of mitosis, post-mitosis fertilization. So that's a nutshell of the pollen cycle. But the thing is, something to bear in mind is that this is done through gymnosperms, seed producing plants. And that allows for the fertilization to occur even in dry or arid conditions. Now, pollen transportation can occur in different routes. It could occur through wind, traveling through air, and even through mammals or insects or animals, right? So think of like dragonflies, think about like squirrels, things like that. So think about these different properties because we're going to talk about them shortly, or some of them at least. And once again, flowers are reproductive, have reproductive shoots that are specialized for the transfer and receipt of pollen. So think about the different diagrams that hopefully you've seen before that really illustrate, like the one I showed in a previous experiment for exercise 2, to highlight, how the pollen transfers and moves towards eventually the ovule for fertilization of eggs. Now, and yes, there's diversity or there's unique traits of these flowers in different methods and different traits that they have overall. But briefly now, we're going to move towards the exercise, and you're working as groups again. However, actually, in this case, you could work as pairs. You have two options. Sorry, this exercise 2, yeah. So in this case, and you also have, I forgot to mention this as well earlier, but if you want to use or look back at any of the ovary slides, pretty much the ones that we've done for exercise 2, you can have a look at them here through the light microscope that you have as well. But for exercise 3, where we're going to spend more of our time, we're going to use the red box for our slide. So you could use and take as, let's see, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there's 12 slides here. So you can work as sets of 2, pairs of 2, side by side. And if you wish, you could use your pollen, the pollen from your flower actually for exercise, from exercise 2, to place them onto the slides that you have from your initial group, right? So the idea is that you're going to place them onto these slides. You can add in the cover slip on top with just mounting. It looks like there is like a mounting slip. Water can also, so water suffices as well. What you'll do is if you want to continue with exercise, if you want to use your slides or make your slides, I'll show you how to do it shortly. But the point is, start off using these ones, and you'll be able to see and create an experiment for yourself using your lab manual. So, and the different components that we've talked about today. What I mean is that, okay, let's say you look at your flower or the pollen grades themselves. You'll see so many different types, perhaps 20 different species, and you'll be able to know the categorizations from your lab manual. But look at now just even maybe 2 different key categories. Some pollen grains are larger than others. Some are perhaps from flowers that are eudicot, others are monocot. Some come from flowers that are insect pollinated and others that are wind pollinated. So they differ in different types of categories. Your aim as sets of 2 are to be able to essentially compare 2 different plant groups and to derive how they differ in their plant pollen grain diameter. So bear that in mind. Your dependent variable, or what you're focusing on to differentiate your plants into 2 groups, is their pollen grain diameters, their lengths. And what you're doing to do this is, just want to see everyone has a light microscope. Okay, so this is a bit different. So to make it easier, rather than working as pairs, work with a person on the opposite side of them. If there's no one on the opposite side, then work as pairs. The reason I say this is because before we used to have the light microscopes on, actually multiple, not mistaken, on both sides. If not, they were oscillating. But the point is that you can work as pairs, and the same continues for the rest of you. Likewise, the 2 can work as pairs, and the rest of you can work as pairs accordingly, right? All the way to the end. If you find that it's if your microscope isn't working or if you're having concerns, just let me know. But the idea is that you're going to do this experiment once again, looking at your pollen grades, let's say from the slides or from the one that I told you earlier. You're pretty much just making a slide. You have a slide in one of the boxes for each block that I mentioned before. And what you're doing is just adding in a few speckles or multiple speckles of you add in your microscope, your slide, add in a few drops of water from the ointment shown across your bench, and then you add in speckles of pollen, so that way with the cover slip on top, you're able to see your pollen grain seeds or your pollen grains, and then measure their diameters. The same occurs with the slides here, but you have more to work with. And then that way, if you use both, then you have 21 species to look at, which is nice. And then so once you're at that stage, right, once you're looking at your slides, then you want to be able to compare, okay, I see different types of grains. What are we going to differentiate or compare as pairs, right? So the idea is that you're going to do this step of comparing the different properties that you'll see in I'm just going to look at the pages of your lab manual that include pages 13 to 16. So look at that for reference for the different categories of those pollen grades that you will find on the slides, and then you'll be able to determine the diameters, which we'll discuss shortly. Any questions? No questions? Yes. We could do that, but the reason why I didn't want to miss a week's lecture on that one. But if you wish to, if you have more space, then it's okay to actually move the microscope. Otherwise, you could just bring in your chairs, which is relatively more easier to do. There is two other things before we end off. The number, so you've imagine, let's say you're focusing on how the different flowers, like flowers with different modes of pollination, have different pollen grain diameters. So let's say that's your research question. Then you could use, once again, I said the slides shown here or the one that you'll make shortly. But then the number of species that you're looking at, look back at your lab manual, you're pretty much using maybe 2 to 3 different types of species, but you have 2 different groups, like a table of 2 categories. So it could be, let's say, insect pollinators versus wind pollinators, or eudicots versus monocots, or large seeds versus small seeds. Then after that, you could put the different types of species you have, but importantly, put in the values that you see from your microscope. And you're using the light microscopes, which are the ones that have stages distinct from the ones that you use for the dissecting microscope. And then lastly, you're going to be measuring those values, and there could be 8 to 10 per group. 5 to 10 per group, max 10, and they need to be consistent for each group. The last final component, and forgive me for the timing, but this should be hopefully understandable. Let's say now you're using your microscope, and when you familiarize yourself with it, you'll notice that you have a ruler on, like an ocular micrometer, an eyepiece micrometer, a ruler on one side. So when you're looking, just try to be able to measure how much a seed is. And when we go to essentially understanding how to measure it, you'll see it in your lab manual, but the point is that, let's say this is as an exercise, 15 OMU, 15 essentially ocular units from your microscope. If you're using, let's say, 10x, what you're going to be doing is just multiplying this calibration factor by how much you see. 15 times 25 is 375 microns, right? And then, and that's pretty much the size of your seed, of your grain. You're going to repeat that process for all of your categories and all of your values per category, such that they're even. And then at the end, you're going to get that mean for each group, and then you're going to derive the other calculations to come. Any questions? Was that a bit exhaustive? Any concerns, questions? Thoughts? Does everyone understand pretty much what needs to be done? Yeah? Okay. So you can go off now. This is just another example, but the point is similar. I would recommend try to go to 40x if you can.
to do the measurements. If you find it easier at 10x, you can also do this as well. But you can go forward, continue as you can for the next, I would say, trying to get as many values in the next 40 minutes. So context for the timing, try to be done by 11:30. Sorry, 12:30. Any other final questions? No? Okay, thank you so much, everyone. You can go straight to grabbing at the period.
No, I don't think it's I put it in this yeah. And I would just use science because that's what I
I'm also going to try to provide updates every 10 minutes. So hopefully just keep an eye on time. I'll try to provide reminders for you all, try to keep you all up to date. If you have any questions, let me know. Once again, you're using the slide box. You don't have to take it from exercise 2. Use the one from the red slide box, not the one from the ovaries. There's a red slide box. I think someone slide from the ovaries. Yeah.
good. Is there like a lot of global?
Anyone took a slide from the ovary? There's like one, like someone took one ovaries to that, and here, that's before
Yeah. There's a lot of good ones. See what those slides.
I just need to know who has the one from the ovaries.
Okay, that's, I think that's a.
Who's taking the slide from the ovaries for exercise 2? Because we're at exercise 3, and I have one remaining slide exercise. So I just need one. So hopefully everyone has a slide now. And if you need, hopefully that should be enough because it has 20 different species. So just bear this in mind, everyone.
The yellow one.
So, let's go back for everyone who's having some difficulty. So let's have a look back at this slide. If you're using the red slide box and you're looking at the slide right now, hopefully all of you, how many of you are able to focus onto the slide? Who's having problems looking at the slide? It's okay. If you're having problems, okay, just let me know. Anyone having problems looking at the microscope? Yeah? Anyone else on this side?
Are you able to focus on the microscope, sorry, the pollen grain through your microscope? No.
Yeah. Alright, what are the difference between these two?
I'm not sure.
So, oh, large, yeah.
All the others.
Yeah, you're right. I told you, large. But I don't think that counts.
That's another. But we're no farther left.
Basically, any different extent.
Down, small squares.
It says pollination.
And then what? Okay,
You can do that. What do you, so what's your name? What do you, what is
We were just looking for.
keep looking, yeah.
Oh, yeah. No, that's right.
I would say really one. So looking at
Oh, yeah.
what is or from what you see right now, which ones and that could be
Yeah.
no problem. So how's everyone doing now in terms of focusing their microscope? Is everyone able to see the slide? Everyone's able to see their slide? Yep. So I apologize if it was too quick for you guys. Let me just tone it down, but I need everyone's attention, right? So now that you focused on your slide, what's next? Next off, you need to look at your manual, look and compare from the different pollen grades that you've seen thus far. Which ones do you want to characterize, or sorry, which ones do you want to compare in diameter? And how you're doing it is based on two different comparing comparing a trait with two different categories. So for example, pollination method. Are the pollen grains that I'm seeing, are they either insect or wind pollinated? Can I compare two different categories like that? So to make it easier for you, this is what you're going to be doing on, I think, in your lab manual, page 11. On page 11, you have the idea of, or sorry, the categories, let's say category A and category B, right? Once again, A and B could be monocot versus eudicot. It could be insect pollinated with pollinated. It could be angiosperm versus gymnosperm. It doesn't matter, so don't worry, you don't have to trivialize on this. So what's important is that once you find out
And everyone, please pay attention. Are we paying attention? You have two different categories now, right? So what you're going to do is the following. You're going to have measured the different pollen grain seeds, or sorry, the diameters of grains for multiple, with multiple values. So you're going to take, let's say this is you focusing on insect versus wind pollinated pollen grains. Let's say the wind one is blue and the red one is insect pollinated. The idea is the following. You're going to be taking different images of the blue ones. That could also include the other big blue ones, like blue-green ones, like here, or there, right? And then the red ones could be like these ones, or the other ones here, right? And so the idea is that they're going to be separated into two different categories. Once you do that, each one is going to be given its own value, right? And you could label it by its species name, which you'll see from pages 13 to 16 of the manual. So once you do that, I believe, so I just put species one for an example, but once you, there is a section for your pollen diameter, right? For your pollen diameter, and sample one is just like value one. So it's like grain one, grain two, grain three for that single category. Species one could be the same species, or ideally you have 2 to 3 species across your entire list. But the idea is that you're doing this and you're determining the size of your grain in microns or micrometers. And you just repeat that process. This could be sample one, sample two, 3, all the way down to 5 to 10 in each category. And they have to be the same. i.e., category A should have 7 greens, and category B should have 7 greens. So the number of wind-pollinated pollen grains you've calculated their sizes for should be, let's say, 7, and the same for insect-pollinated pollen grains, which would be 7. It could be 6, 6, 5, 5, but don't go over 10. And hopefully by that time, even if you can't, like if it helps, try to essentially write as many values as you can just for those categories to align up, to match up. And don't worry too much about the species. So long as you have, let's say, 2 to 3 here for each category and 5 to 10 of the number of grains, then that should be okay. Any questions? Final questions on that note? No? So is that okay? Any questions? Confused? How many of you are confused? Yep, okay. That's okay. I don't mind. So I'll come help if there's concerns of individual focus. But in the meantime, look back into, so I just touched upon this, but look at this slide to be able to now compare the sizes of your pollen grains, right? So let's say, and just make sure everyone you're paying attention. I'll come back and make mention to this, but now just try to count pollen grains.
So these are all the blue ones. There's so many blue ones. There's so many five. Yeah, what you will make it the real now. Yeah, yeah.
Everything okay? So just to reiterate this, you can do even 7. My recommendation is 7 to 8 to get diversity around 7 to 8 here. 7 to 8 here. Just to make sure that you're consistent. If you have 7 here, you have 7 here, 8 here, 8 here. And the number of species could be 2 to 3 in each. Don't worry too much about it, but the idea is that you really need a good number of samples and a number of species to compare your findings.
Yeah, that's the photo, like we use you have to find those one. So look at 10. So go to a different one. Yeah. I guess. So, I'm sure some of them. Oh, yeah. I think we can. Yeah. Yeah. Okay, we're just looking at yeah. So yeah, you just go all the way. Yeah.
It's like,
Yeah. That's
wait, yeah.
I'm gonna ask each set of pairs, how many pollen grains have you measured so far? So I'm gonna start from your end. How many have you counted? How many? What do you mean, like, number of pollen grains? Like, how many boxes have you filled up so far? I'm actually have one thing that how many have some values have you collected? Four. One box.
I'm going to find purple ones. Things like powder. Oh, wait, small, like, yeah, ours are really easy. No, you.
Everything okay?
Oh,
Here's your Before 0.6, to make it as efficient as possible, try to count as many pollen trees as you can and try for a single category. Then go to the next one. Because if you can reach out to survive in one, move to the next one, and if it helps you everyone, just to make sure everyone's clear for timing, try to finish 5 during at minimum for one category, 5, 12, that's to be acceptable for time. On screen, measure the size, write it down, and that should take 1 minute per on breed. And then you can repeat it for the next category.
Yeah.
Oh, this one. Right. Orange.
And these. Yeah. Wait, so these are small. Yeah. Yeah. They were. Yeah. The bottom three. Yeah. The right. Oh, we know that here. Yeah.
Yeah. Well, that's so that's oh yeah. That's what we have to do.
How many pollen grains do we have so far? Yes. 6? Okay. How many do we have from your side? Okay, that's good. Okay. Your group 10 for each. Okay, and your side? Forgive me, I forgot. How much? 5, 6? 5. Your side. Sorry.
Yeah. You want to say the other thing, so I can like see the number in the that makes sense to everyone. Right. Yeah, right. 25, 25. Okay, so what do you Yeah.
So the
Yeah. You guys are gonna be a lot of work. You could actually build it out. Yeah. Yeah. Oh wow, you did the same. That's very bad.
So what do you guys think? So everyone, you have about 2 more minutes. 2 more minutes to get more values. So remember what my advice is, that you try to get as many data points for the measurements that you have remaining. Please make sure that you're as efficient as you can be, because shortly I'm going to talk about stats and how you're going to be able to create your graphs for your post lab. So bear that in mind. Thanks, everyone.
I would just say keep what we have and just check them out again. Yes. And I don't want to explain the big area. Yeah. That should be fine. I think so, yeah. I don't know about the software, but yeah, it's the same difference. You're about 12.5 away. Budget one. Yeah. You share the stuff that we have there. Like, they have the.
So, everyone, I just want to make sure we're okay for timing. And we're going to be moving actually now to stats. But before I do so, can I make sure everyone has done at least 10 samples? Who has not done 10 samples as of yet? Okay, how many have you done? Okay, okay. How many of you have not done 10 samples?
Yeah.
Okay. How many do you have remaining? 4? Okay. So my anticipation is that as I continue with stats, have one of you try to focus in with regards to what I'm about to say, because this is a bit more heavy and is involved for your post lab. So just bear this in mind. We talked about, determining the diameter, and pretty much you're doing the calibration factor of what I've said earlier. Let's say if your size of this pollen grain is 0.5 OU or OCU or OMU as ocular micrometers or micro units, if you multiply that by the calibration factor, which is 25, that's 12.25 microns, also known as micrometers. So now when it comes to the stats, and normally when you're doing this, I know we're high paced right now, but normally when you're doing this type of experiment, you do start off with observing your biological material, your specimens, your species at large, and then you make it a hypothesis. So for example, when you're saying, what is your goal really? Comparing your independent and dependent variable. Your independent variable is the one of the two categories, or essentially the main parameter that you're exploring, i.e. pollination method, or eudicote versus monocot, i.e. the morphology of your flower, or the, I said pollination method, or the type of your flower, i.e. angiosperm versus gymnosperm. So those are three main parameters to explore as independent variables. But then the categories are the ones that I mentioned earlier multiple times. And what you're doing is the following. When you're saying you have a hypothesis, you're saying, let's say here, your hypothesis is a specific conclusion. And I'm not saying as a concluding statement, but it's an inference that you're making depending on your values. So for example, you're going to say for a given hypothesis that you expect there to be a, and this is where the difference comes in. I'm just going to put it square for a reason, but a difference between the categories of your independent variable. So we'll just say categories of your, and I'm just going to put this all as one line. Reason being is because if this is your, so once again, your parameter, the categories are between pretty much of your parameter, still the same. This could be many types for each and every pair of you. So for example, if this is a null hypothesis, actually, if it's different colors, if it's null, this is going to be in black, then there's no difference, right? Actually, yeah, there would be no difference. And if it's your alternative, right, which is going to be a bit larger here, but alternative, meaning what you're going to explore, that means that you expect there to be a significant difference, right? So it will be a significant difference between the two categories of your parameter. So a significant difference in what? In your pollen grain diameter, for example, in comparing your pollination method. The two categories of your pollination method could be insect versus wind. It could be insect versus mammal, etc. Or it could be eudicots versus monocots. It could be angiosperms versus gymnosperms. So think about those as your parameters, but connect them back to what you're measuring, your pollen grain diameter. So that's important to bear in mind. Any questions on that? Okay, so the next step is calculating your mean, variance, standard deviation, and 95% confidence intervals. I know that sounds like a lot, but the lab manual makes it quite explanatory. Your mean, we likely know this, that you pretty much group up all of the calibrated micrometer sizes or diameters of your pollen grains. For one category, you sum them up, divide them by the number of pollen grains you measured. That's your mean for one, and the mean for another, for the second category. The variance is also provided as a formula in your lab manual, and the same for the standard deviation. And then thereafter, you're calculating your 95% confidence interval. That indicates to you how confident you are that your mean is the observed mean is what you would have expected and what is true essentially. So hypothetically, if your 95% confidence interval is between 30 and 40 microns, then you're confident at least at 95% of you to keep on like essentially doing these measurements, you would get your values somewhere in that range. And likely having a mean around 35 microns. So that's pretty much what your confidence interval is. And then you're going to be using your mean. So think about it like this. You've calculated your mean now, hopefully from your lab manual. You could do this even now or in a few seconds or minutes after this brief presentation. And then you include the 95% confidence interval that you could calculate afterwards today or shortly. And then you interpret these two together such that you have the mean right here shown in blue and the confidence intervals shown separately for both categories. Make sure that, as you can see here, they're labeled the x-axis and y-axis with the units. And by the way, we're not doing petal size, we're using pollen grain diameters in microns, so note the difference. Then, when you see and compare the confidence intervals, you notice that they don't overlap. That means that there's a significant difference, right? Because if obviously you notice that your mean is away from the other means category or the other category's mean, that signifies to you outside of those confidence intervals that they are distinct from one another, right? So the pollen grain diameters for monocots versus eudicots or insect versus wind-pollinated flowers could be different or they could overlap. And if they overlap, then you can't reject the null hypothesis, right? But if they do overlap, then if they overlap, you can't reject it. But if they don't overlap, then you can reject it. And so that's pretty much a bit about stats. Any final questions on that? No? Okay. Cleanup is pretty much what we have now. A lot of things should be already cleaned up from what your benches look like. However, I do want everyone to pay attention to the following. When you're done, make sure you place back all the slides into the red box. I do want us to be done in 5 minutes at max. So this is your time that if you haven't collected when I was mentioning the diameters during the presentation, this is your time to complete it and then start cleaning up. What you could do is place the slides into the red box, clean up your razor blade, placing it back into the small petri dish like this, close it up, place it inside your white bin. The same occurs for your petri dish with your pollen grains. If you made a slide, pretty much with the water that I mentioned earlier, but you clean it up, rinsing it out once again, and then make sure all your razor blades are back where they were placed, including the styrofoam, and your slides can be placed into the glass bin. So that should be everything. Thanks a lot, everyone, and look forward to more amazing practice. Once you're officially done with the practical, please remain seated and look at the slide, or I'll mention more details on the slide to come. Thank you, everyone. Let's try to be done by 12:48, everything, including our cleanup. For any flowers, you can place them into the garbage. I honestly would prefer that we put them in like an organic bin. but hopefully they'll be disposed appropriately, or hopefully they'll grow up again. So just bear in mind any other things that you have, pollen grain residue on your Petri dish, you wash it off. Make sure that you clean up all of your bench items. The next class will be coming shortly, so make sure that you clean up your bench accordingly. You can shut off your microscope as well. You can lower down the stage, and you should be able to clean up. And you should be done actually very soon. So, pretty sure we have to. Yeah. So no one leaves the lab until I tell you the final instructions for the post lab. Very important because due tomorrow I want here. They have to submit it, and I want you to install agent, and don't worry about your conclusion. As long as you're all done. I have to go. I'm second here. Place all the belonging slides, all those slides need to be placed back. Not very well. So we're not leaving as of yet. You can wash your hands. You can pack up your bags if you wish. But I just want to make sure you all are complete. And I believe there's one or two more slides that are still missing. So now that we're done, and make sure that you set off your microscope. You don't want it to be running on still. Make sure your bench is clean. You can use the paper towels as well. But there is the post lab that you all need to be aware of. Anyone needs to drink for any moment in time during the last stage, partially just let me know. You can just use the washroom. Just keep me in the loop. But don't worry about this. Just bear in mind that you're focusing. The post lab includes at least three components. There's more that's in your post lab, but these are the ones that you need to prep for in advance. You're going to be making a graph, the graph that I showed you earlier, with the details of your means for the two categories, the labels of your axes and the units, of course, and also your figure caption. Bear that in mind, as well as making the confidence intervals I mentioned from the end of your lab manual. You're going to upload them, you're going to plot them. Please pay attention. So I'm not trying to like disillude anyone, but please, I want to make sure you all are aware that you're going to need to be able to complete this from 24 hours from now. So before 1 p.m. of Thursday, inshallah. And then after that, that's your results section. Following that, you're going to have your concluding statement where you're going to talk about your results and your alternative hypotheses. So you're going to have a lot of data that you have to upload from your lab manual, what you've already uncovered, but make sure you're able to describe these details with specificity. and as much comprehensive knowledge that you have from the null hypothesis, alternative hypothesis, all the stats that I've mentioned thus far. And then the last or one of the final components is your limitations from your study. So what did you find out from your results? What did you see from your comparison of the pollen grain diameters? What limitations could you consider? How many seeds or pollen grains did you actually measure out of the many that you could have explored? How many of them were easy to determine? How many of them were confusing to describe? How many of them actually reliably show the diameter of your pollen grains? Because they could be oriented like this. They could be oriented like a circle. And it may be easier, let's say this is your grid, or let's say this is what you're seeing from the slide of your field of view. This could be your pollen grain, and this is what you see. But your pollen grain could actually be like this, or squiggly, right? Or like having different morphologies. So the idea is that you're looking only at one angle. So just bear that in mind, or one field of view. So bear that in mind when you're coming up with your limitations, and you only just need to refer to one, but then describe in depth how you would redesign your experiment to resolve that limitation. That's it for the lab. My advice for you all is just to prep for the post-lab or your next week's lab, 2 weeks from now, and all the best for everyone. Thank you so much. Feel free to wash your hands and have a great rest of your day. Thank you, everyone.
You don't like null hypothesis rules on it. The pollen size does not change between the medium-sized pollen grains, whether or not they're transported by wind or sun.
That's good. That's good. Nothing here. I would just do because you added an additional feature, because technically you would only be focusing on either or. So one pollination method rather than the size. It's okay as it is. You don't have to change anything if that's the case for all of them, that they're all medium size. So that's okay. It's good as it is. And then you just do the same for the alternative. But
I just want specific.
exactly. Thank you. No problem.
So,
Yeah.
I have a picture. So, the Excel that you're showing us with that, with like that, are we gonna put it into like the first file? We have a question where we can have the data.
You're gonna have to put the graph and you're gonna upload it onto the person.
Okay.
Your post lab is also on Quirkus. Just bear that in mind, everyone.
Okay. That's all. Thank you.
No problem. What's wrong?
It's not.
Yeah. I just had a question.
No, don't know.
I would like to. Is that your computer, by the way?
My turn.
Okay, go ahead. No problem.
Hey, you don't mind if I set up here?
Of course,
Yeah.
of course. I'm just gonna clean up some things, but go ahead. No problem. What's your name, by the way?
Kian, nice to meet you.
Kian?
Sorry, Mohammed?
Yeah.
Nice to meet you.
What's your program?
Like supervisor
Or like, are you an undergraduate school?
I'm a second year PhD student. How about you?
I graduated.
Oh, I graduated already.
But I hope you're enjoying your midst of the thesis preparation. Thank you. No problem.
I'm trying to make this right.
Yeah. Okay. Yes, you have a question.
I don't know exactly 10, just do first one.
But if there's any or any conversation, try to grab the phone from them. If they have when exactly. Yeah. Just one more data. You would need like the one on yeah. No problem. Just I would say be mindful of the parameters that you use, as pollen grain diameter, not the petal size, petal size. You just bear that in mind, but it's very similar in format. No problem. I find that it was really stressful. That's great to hear.
They're very calm person. It reminds me of myself a lot. So that's why I'm over and sort of talk.
That's nice. I really appreciate it. They give me because I have multiple students. I need to remember your names. Josh and Chan, right? Perfect. Okay. Thanks a lot.
No worries.
I hope you continue to enjoy the lab.
Oh, no, I definitely am. You have, I think what people things go wrong and then it's like, oh, it's bad, right?
Thank you so much. Everyone else okay? Yep. Okay, please, one last thing important to go. No problem. When you're done, just don't forget to wash your hands. And then you could leave. Thank you. Okay. Everything okay? No problem. Take care, God. Everything okay? Okay. Have a great day. Alaykum wa rahmatullahi wa barakatuh. So, alhamdulillah, Alameen, Rabb al just finished the first week of looking forward to inshallah ta'ala helping students to come closer to learning about Allah through the noble practicals of biology. So inshallah ta'ala, of course. So take care, everyone. Looking forward to seeing you all soon. Barakallahu feekum. سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر جزاكم الله خيرا كثيرا والسلام عليكم ورحمة الله وبركاته.